My name is Tan Zhao. I'm an applied research scientist from NVIDIA, and I'm happy to host today's MICCAI industrial talk from Dr. Fernando Pérez-García. Fernando is a senior research machine learning engineer at Microsoft Health Futures. His work within the Biomedical Imaging group focuses on translating artificial intelligence research into clinical practice. Prior to joining Microsoft, he worked at the Center for Neuroimaging Research of the Paris Brain Institute before moving on to UCL and King's College London for his PhD in medical imaging, where he investigated the potential of artificial intelligence to improve the treatment of epilepsy. Fernando is a strong supporter of open source software and a recurrent contributor to multiple open source projects. Welcome.
Thank you. Thank you very much. Thanks for the opportunity for letting me present our research in this MICCAI. This is exciting for me. So I'm going to discuss a little bit. I'm going to present the latest research of our team. We've been working on multimodal tools. I'll explain what I mean later by that, to improve, to help radiologists do the job, basically. So we are the Biomedical Imaging group in Cambridge, UK. We are part of the broader group called Microsoft Research Health Futures. And in the Biomedical Imaging group is a multidisciplinary team. We have experts in machine learning, medical imaging. We work with radiologists, expert in human-computer interaction. And an example of one of our recent projects that has been successfully deployed is INRI. So INRI is an open source software library used for segmentation of 3D images to support a radiologist so they can do the job faster. And the team not only built the software and trained the models on the cloud, but build the full infrastructure so that as soon as the images are acquired in the hospital, they are sent to the cloud, they are segmented, they're processed, and they're sent back to the radiologist's console for radiotherapy planning. And this is actually now implemented in all hospitals, large hospitals in the Cambridge area. They use INRI trained with data from their own hospitals to improve the radiotherapy treatment, the radiotherapy planning. I think 13 times faster is the numbers that we have in our INRI paper. So it really makes everything much more efficient for them. So we'll discuss here in this talk a bit of our recent research. We have multiple papers about multimodal radiology, some about self-supervised learning, contrastive learning. We assess GPT-4 capabilities in radiology. But here in this talk, I'm going to focus on these three papers. So I'll go one by one. The first is MAIRA. So MAIRA is our project. Project MAIRA stands for Multimodal AI for Radiology Applications. And again, it's all about creating highly capable large AI models that assist radiologists to do the job more efficiently and more accurately and more safely, basically. Okay, so we have a document on arXiv that you can check. You can just Google the name of the paper and you'll find it if you want all the detailed information. Because I don't have a lot of time, I will go quite fast through some of the bits of this research. So this is normally the images at the top, and the text on the right is what a radiologist that needs to assess an image would see in their console. And they would be tasked to writing the text on the bottom left. Given an indication, they would write, for example, a text with findings, which is basically a list of very descriptive sentences about what's present and sometimes also what's not present in the image. And typically I've highlighted only these bits because this is what typically these models are trained with. So instead of using information from prior studies and different views, like for example, lateral view instead of just the frontal view, these models are typically just given the frontal view and then the findings. And this is actually quite challenging because in the findings, sometimes we have sentences that are actually comparing to prior studies or comparing to other views. So we need to be very careful with this. So why do we want large language models? I think it's clear that they've been very powerful recently. These are models that are trying to generate, they're generative models trying to generate text. They are sometimes massive, trained with a lot of data. You're probably familiar with ChatGPT, GPT-4, which is used in this example, or Claude, Llama, etc. And we see that these foundation models have a lot of potential uses in them, a lot of different domains. We actually probed the capabilities of GPT-4 for radiology applications, and we wrote this the paper that is shown at the bottom, and we discovered that they actually encode quite a lot of already information about radiology. So we thought it's a very promising area of research leveraging these models for within our domain. And in our case, we are using multimodal models, multimodal language models, I guess. In this case, maybe for this audience, just to be clear here, multimodal doesn't mean different modalities of medical images like MRI, CT, or T1, T2. We just mean image and text instead of only text, basically. And you might know that there are now some very powerful models out there that have image understanding capabilities like GPT-4V, Cosmos, etc. So they can not only caption an image, but one can also have a conversation about what's going on in the image. Just looking at the Q&A in case there are questions. There are other multimodal capabilities that these models can potentially present. For example, they can output not just text, but bounding boxes in form of text, but they can be converted into bounding boxes, or with some tricks, also segmentation masks. I'm not going to discuss this, but I think it's good to keep it in mind because these are potentially very important applications of language models for this domain as well. So what we do in MAIRA is we leverage these kind of models to generate reports of the medical images. So we give one image, one frontal image of a current study, and if we have one, then we give an indication, meaning what we are interested looking at from this image, given the history or given what's happened recently. And this is strong challenging because it requires strong domain-specific image understanding. This is what we've been exploring in MAIRA, and this is the paper that we've written. So MAIRA is based on LAVA. If you are not familiar with LAVA, it's basically an architecture composed of multiple blocks. Hopefully you can see my mouse. I don't know. But what we need in LAVA is or here in MAIRA is a strong image encoder, and I will talk about the image encoder later. The input is basically the image information and the text information, if present, the indication. If the indication is not present, we just pass the prompt asking to provide the findings. And the elements of this is the strong image encoder. We'll see how important it is to have a domain-specific image encoder. I will talk more about the encoder later. We need an adapter, a projector that takes image embeddings into the space of the language model, and then we need this large language model. In our case, we use Vicuna. We use a 7 billion image, so relatively small, and we fine-tune it for reports. I see a question about focusing on chest X-rays and whether we've considered other modalities like MR for brain, PET, or CTs. And this is a proof of concept that we do in MAIRA. So for now, we're focusing on chest X-rays, but we are, of course, open to exploring actually more challenging modalities such as MR or CT. Yeah. But for now, this is all about chest X-rays. So we use MIMIC, probably the most popular data set of chest X-ray with reports. It has about more than 300,000 images, and there are 130,000 image report pairs, and we use only the frontal images, as I said before. And from their reports, we only take the findings as the target generation and then the indication if present. Just a little note, a little trick that we did is we augmented some of the text, because sometimes the models overfit to specific ways of phrasing things. For example, we might say that the cardiac silhouette is mildly enlarged. Sorry, not that. We may say instead of there is no consolidation, we can say no consolidation is observed. So these little changes are cheap to run, because we can use older models instead of GPT-4 that are faster and cheaper. And then a couple more details. We used DeepSpeed, the DeepSpeed library that partitions the weights, optimizers, and the gradients across GPUs. It takes a couple of days to train this on one node with 4 A100 GPUs. We use some optimizations like brain floating 16 or flash attention, and we keep the image encoder frozen. We only fine-tune the projector, the adapter, and then the LLM. So I'm not going to go through a lot of we have many results in the paper. I'm just going to talk a little bit about the challenges actually behind evaluating these models, and then I'll show actually some actual results. So it is challenging because the output of these models is not just a classification, right? It's not 0, 1. So how do we know that a text is correct? Typically, we have a reference text, like the ground truth, or the human-generated text that then we compare with the predicted model. But this is really not trivial at all. First, the sentences can be in different orders. There might be some synonyms, things expressed in different ways. So this is very challenging. For example, we can see sometimes errors, missing words, or let's say, I don't want to say hallucinated words, but let's say words that were not present in the original report that actually are not important. So how do we know which errors do we care about? For example, sometimes this might happen. In the original report, we have that the heart is top normal, meaning it's still normal, but almost it's large enough that it's almost not normal. And maybe the prediction says mildly enlarged, which means something very, very similar. One could argue. So it's probably not something that we care too much about. And then it might be that sometimes the original reports are actually wrong. So this is a label noise problem that happens sometimes. And something even worse is that maybe the output of the model is actually very detailed, but the ground truth is actually just saying basically there's nothing wrong, or there's nothing interesting in this image. So is the model wrong? Probably not, or maybe not. But the metrics, if we use traditional metrics, or if we are not careful with the metrics, we might think that this is a disaster. So an idea is to use radiology-specific evaluation metrics instead of more standard text metrics that look at precision and recall of words without looking at what those words are. We may want to look at specific metrics that I have developed for radiology. So I'm not going to go through all of this, but I'm just going to show, for example, how the Checksbird labeler is used for the Checksbird matrix. The idea is to extract only from text only the presence or absence of a certain number of pathologies, typically in this case I think 14. So we just check whether they are mentioned or not mentioned in the target and the prediction, and then we can just use standard classification metrics for each class, like precision and recall. And typically we aggregate these across the 14 classes. We call this macro F1 14. And this is seen quite often in the literature. So something we did our best. The team works a lot on evaluation to be fair in the comparisons with the state of the art. But the test sets vary a lot. It's quite hard to match test sets in the literature, but we did our best. We see that MAIRA is the best on lexical metrics. So what I mentioned before about classic metrics used for text generation. And then if we can trust our results, it's also difficult because our model sometimes, for example, this model from 2 and others is about 1 order of magnitude larger than ours. And the test set is quite different, but we did our best. And if we trust these metrics, it seems that our results are similar to the state of the art as well. So we are quite happy. So some conclusions. What did we learn from this? We learned that a relatively simple architecture can do pretty well. As I mentioned before, that our language model is much smaller than many others in the literature, instead state of the art. We saw, and I'll show a bit of this later, that having a radiology-specific image model is crucial. I didn't show results about this, but using the indication as a part of the prompt, the indication of the report is very important. And then I didn't mention this either, but different findings, some findings are much harder to detect than others. And then for next work, this is what I presented is MAIRA 1. That means there will be a MAIRA 2. And so we are aiming to use prior images. I talked about this at the beginning of this section. We need to, as a community, we need to get better at evaluation and be more transparent with our splits and with the way we compute our metrics. And then we need, as usual, more diverse data sets. That's all I have about MAIRA 1. I will keep an eye on the Q&A. I won't be able to reply to everything. I see some interesting questions about, for example, using structured reports, I think. And this is something that our team is very interested in as well. And I think we will probably be looking at that in the future. I am more familiar with the imaging side of things, which is what I will present next. I see questions about augmentation, some training details. I'm happy to reply to this offline as well. And then at the end, I'll take some time to see if I can reply to more of this. So I'm going to move forward for now and present, talk about RAD-DINO, which is the image encoder that we've been working on for MAIRA. So I said before that for MAIRA, we need a strong image encoder. We'll see how we actually created that. Typically, image encoders for chest X-rays the recent ones have been trained using contrastive learning. So typically clip models where images and text embeddings are aligned into the same or push closer in the respective or in the common latent space. So here we have an image encoder and we have a text encoder, and we want the representations of the image patches and the text tokens to be close in the latent space. And the hope is that then we can leverage the image encoder that should be able to extract semantically meaningful information from the images, and then leverage it for any application that we want, basically. However, in a domain, there are a couple of really important challenges. One is that sometimes in the reports, we just see basically the reports say no findings, there's nothing wrong. Typically, it's something like no acute cardiopulmonary findings. And what happens is that images that are actually visually very different are pushed towards the same latent point, the same point in the latent space. For example, let's say we have a data set with some animals and their corresponding text captions, text labels, parrot, giraffe, and then we have other images that are not animals, and the label just says, this is not an animal. Because the text labels are the same, these images will be pushed to the same place, and even though they're actually very, very different, they contain very different information. So this is not ideal. So this is an issue related to using contrastive learning or clip models for chest X-rays. Another problem is that in the reports, so the reports talk about the image, but they don't fully describe the image. And also, even more importantly, sometimes they say what's not in the image, which is something that is not typically seen in the general domain. You wouldn't find images with captions saying, this is not a dog, there is no dog in this image. So that is, it's quite a lot of noise that happens while training these models. And there's a lot of information about the patient that could be extracted from the image, but these image encoders probably tend to ignore because they are just not present in the signal that they get from the text reports that are used during training. And we'll show some quantitative analysis of this hypothesis. And another very important problem is that we want paired X-rays, so pairs of images and reports to train these models, but actually there are not that many. It's much easier to find data sets that have only chest X-rays without reports than with images that have an associated report. So we would like to be able to leverage all of the data, not just the subset that has in general domain would be a caption. For us is a report. So the solution for this is trainings models using self-supervised learning, but only with images, to have a image encoder for MAIRA. This is not my coolest slide, but I think you get what I mean. And just to talk a little bit about self-supervised training, maybe in 5 minutes. Typically, historically, there's been self-supervised learning methods have been either generative, where we mask part of the image and we have to generate. We ask the model to encode the masked image, and we ask a decoder to generate what was missing. Or there are, for example, discriminative techniques where, so this is the, we can do puzzles, we can do rotations, but the idea is always the same. The hope is that the encoder will be able to have a meaningful semantic representation of the image. So whatever we train, we just keep the encoder, and we ditch the rest for application. What we use in this in our method is a joint embedding network. So these are Siamese networks. It's basically based in this philosophy. So here we have BYOL. We have a, I think, student, it might be called differently here, but normally, here, online network and a target network, also sometimes called student and teacher network. And what typically happens is that during training, we take an image, we apply 2 different transformations, and then we ask the student to match the representations of the teacher, or the online and the target, because normally, we give less information to the student than to the teacher, and then often what we do is that we backpropagate only through the student or the online network, and the target or teacher network in our method is just an exponential moving average of the weights of the teacher. So related to our method, and then we keep the encoder of the teacher for later representation. Something that is also very relevant for us is masked image modeling. So similarly to masked language modeling, which came before, in masked image modeling, in this case we have the masked encoder. Some patches of the image are masked, and then an encoder is given either, depending on the method, the whole image with special tokens for the mask, or just the unmasked tokens. And then we ask our decoder to generate, in this case, generate pixels of the original image. It might be that we ask to generate just a latent representation, as we'll see later. And then, as usual, we just keep the encoder for whatever task we want to do after. So our method is built on top of DINO v2, which is also very, very similar to iBot. So this is iBot, and basically we're having a mix of two different things. First, we have an image. Let's go back. We take an image. We apply. We apply some augmentation to it. We have two views, maybe. And then we have local crops with small field of view and global crops. And the global crops, so the larger field of view, we contain more information about the image, are passed to the teacher network. Whereas the smaller crops with smaller field of views that have less information are passed to the student. And then basically we ask the student to match the representation given by the teacher network. And as I was saying before, the teacher is updated using the latest versions of the student. So we do that for global information using this special classification or class token. But we also want to have strong local representation. So we use masked image modeling. So the student is given masked images, and then we want the representations of the student to match the representation of the teacher, but in latent space. This is related to masked encoders, but again, this is in latent space, and as we'll see, this helps for dense prediction tasks like segmentation, or in the general domain, things like depth prediction. Then we keep the teacher encoder only, and in this case, this is what we keep, and this is what we put into MAIRA model. So how did we do this in our domain? We took a bunch of data sets. Some of them have reports, some don't, but in this case we don't care about that. We use both frontals and laterals. Because we might want later on our model to be able to encode lateral images as well for different tasks. We have about 800,000 images in total. We adapted some of the hyperparameters and the augmentations for our domain. For example, it might be that we don't want to do horizontal flipping, given that the heart is almost always on the left. We don't want to do maybe some color jittering, as our images are not RGB. We conducted some ablations to understand the contribution of each of the features of the method. For example, checking whether masked image modeling helps or not. We'll see that later. Our images have the image size is 518. In the latent space is 37 by 37. We use batches of size 14, and the encoder is a VAT base, should say 14, not 16, of 86 million parameters. So I guess relatively small nowadays. Okay, so some qualitative results. What we do here is we pick a point, for example, we look at the representation, the latent representation of the patch in the center of the blue circle. And then we took a different image from a different patient, and we look at the distance or the similarity between the latent representation of the point that we picked from our original image to the representation of all the patches in the other image. And we see that the model encodes strong semantic anatomy information. For example, here we click on the blue and we see that the right hilum is actually correctly highlighted in the other image. We do that for, we did this for a couple of images. We see this is color coded. So for example, the red one is the last one, the clavicle. We see that the clavicle is highlighted, the one from the same side, but the one from the other side is also highlighted. So this is nice. But my favorite figure is this one, because we see that we give, again, these are different patients. And when we select a patch containing a pathology, the model correctly identifies, or let's just say more scientifically, that the similarity between the representations of the picked patch and the representations of the patches of the target image where the pathology is present is very strong. So, and we see that in the image on the right as well, where we click on the lung nodule, and the model correctly identifies a lung nodule on the other in the other lung on the other image. And I think this is very exciting because it shows how semantically meaningful the representations of this encoder are. We have also some quantitative results. So our model is very strong for classification, pathology classification, especially for rare diseases in our data sets like tuberculosis or for subtle pathologies. And we see that it scales well with data. So this is promising because this is one of the goals of the paper, right? Trying to make sure that we can use more and more data, even if we don't have the reports. And the performance just keeps increasing. We also checked for information that is not in the reports, and we indeed do better than methods, contrastive clip methods that use the report signal to train. So DINO, RAD-DINO is much better at predicting things like the sex of the patient or the age, which is not present typically in the reports. And just to make sure that we're not doing better just because we're using higher resolution, we train DINO at different resolutions, and we see that it does better at all resolutions compared to other models. For segmentation, it also works better than state-of-the-art models, and we see also we ablated to make sure that masked image modeling works, and indeed is very, very helpful. And we see that also RAD-DINO is data efficient. So we just train a linear classifier on top of the, and then upsample the representations. And we see that we don't need much data to do a pretty good job compared to other models. So here we have that basic setting is the green. And this is just an example that we can actually push that performance quite high if we use stronger decoders for segmentation. A little bit of report generation. Here, just a comparison with general domain image encoders that have been trained on much larger data sets. And we see that the importance of RAD-DINO compared to CLIP in a different, so on the left we have lexical metrics, but then we have radiology metrics like the ones I mentioned before, and we see that RAD-DINO improves results for everything anyway. So conclusion of this, we have a model that doesn't need text supervision, so we can just rely on the many images that are available. We see that it works pretty well for classification, for segmentation, for report generation, basically for everything we tried. We can use a lot of data. As I say, we don't need to rely on smaller data sets that contain reports. We can just take more and more images, and it opens, it paves the way to train similarly image encoders for foundation biomedical MLMs, I guess, multimodal language models like MAIRA. Okay, I'll stop there. Again, I'll try to take a look maybe at the end, the questions. Hopefully we have 10 or 15 more minutes to present one more paper. So I'll talk a little bit about RadEdit. This is our most recent work, where we use generative models to stress test other models. So we know, okay, we have these shiny models, but we know they're all a little bit wrong. Some are useful, and this is typically because the data sets that we use to train are biased, right? But we want to know how wrong our data sets are. Sometimes this is actually impossible or very expensive. I introduce an example of this. So this is a chest X-ray with pneumothorax. So that means the thorax has been punctured, probably maybe by a broken rib, and then air comes maybe out of the lung. So there is air between the lung and the thoracic, and within the thorax, basically. So we see that on the left, we see that everything is lung. On the right, we see lung closer to the spine, but then I don't know if you see my mouse, but then less medially, more laterally, we see that there's air behind the ribs. So that's the pneumothorax. And when there's pneumothorax, there's typically a chest drain, a tube that is introduced to suck the air out. This means the consequence of this is that typically pneumothorax and tubes tend to co-occur in the images because there is pneumothorax. Then you put the tube in, then you acquire the image to make sure that it's in the right place, for example, or whether it's the pneumothorax is still present or not. And I'm showing very easy, very clear examples here. But pneumothorax is actually typically very hard to detect, at least by me, and I think by the machines as well. Whereas tubes are quite easy to see, as you can see on the image here. So what happens is, let's say we train a classifier to detect pneumothorax from images that look like what we mentioned, right? If there's no pneumothorax, probably there's no tube. If there's pneumothorax, there's probably a tube. What happens there is that we're happy because our model has done very well. Our metrics are really good. But we all of our images or most have no pneumothorax and no tubes, or pneumothorax and tubes. And we actually don't know how the model would do with images with tubes but no pneumothorax, and vice versa. But these images or these these cases basically sometimes are just very expensive to acquire or just impossible. So we would like a method that allows us to simulate these cases to evaluate our models more thoroughly. So the aim is to have a model that can edit images or generate images somehow that can be used for stress testing. For example, here we say remove pneumothorax, but we want the tube to stay. That way we can test our model and see if it's looking at pneumothorax or it's looking at tube. But this is challenging. So for example, other methods are state-of-the-art. We'll do things like edit the whole image. We maybe just want to remove the pneumothorax, and the method might or might not remove it, but typically we'll just edit everything else, and that's not necessarily what we want. We want minimal changes. Or because this is something that happens a lot as well, is that because the generative model has learned the confounders in the original training data, if you ask it to remove the pneumothorax, it will also remove the tubes. Or if you ask it to remove the tubes, it will remove the pneumothorax as well. So that is really not what we want. So we therefore introduce a method, RadEdit, that uses inclusion and exclusion masks to, for example, in this case, remove the pneumothorax only. It's not very clear in this image, but I promise it has removed it, whereas the tube actually has been kept. despite the fact that there are confounders in the original training set. So I'll briefly discuss the methods. I'll talk a little bit more about the experiments later. So the first thing that we need is to train a strong generative model to generate images from reports. For that, we used the popular latent diffusion models. It's not very important how this works. The diffusion process happens in the latent space of the of a variational autoencoder. We use an BAE, an autoencoder that was trained on general domain. It seemed to work quite well for our X-rays, and then we trained the denoising unit using reports for conditioning and using our chest X-rays for training the denoiser. And just a couple of qualitative examples of this. We need a model, a generative model that actually has high fidelity, so it generates what we ask it to generate, right? So here's an example. These prompts are from the Röntgen paper. We see that if we ask it to generate an image without pleural effusion, that we don't see any pleural effusion. If we say bilateral pleural effusions, we see effusion, meaning there is liquid in the tissue. We see it in both sides. We can control for the site. So on the left, we see pleural effusion on the right lung, which is on the left side of the image. And we can say whether it's small or big, and the same for the other one. So basically, the image fidelity is high, meaning it's generating what we are asking it to generate. But also, we don't want it to generate a high fidelity image that looks the same every single time. So we want them to have high diversity as well. So at the top, we have asked it to generate a big pleural effusion, big right pleural effusion. We see that it does. So all of the images have big pleural effusions on the right side, but they're all different. And similarly, at the bottom, we have the same thing for the left side. So we're happy with this generative model. So the inputs for our algorithm are an image, a target prompt. So basically, how, with natural language, what we want to achieve. So for example, removing the pneumothorax, say no pneumothorax, which is what we see in the reports. And then we have two masks. We need an exclusion mask and a keep. So basically part of the image that we want to keep as it is, although we have soft constraints for this. And then part of the image that we really want to change. And I'm going to go through 3 experiments. I think I have time for this. So what we do is we simulate 3 types of dataset shifts. And for each type, we evaluate a weak predictor that has been trained on biased data, and I will talk a bit more about that in a second. And also a strong predictor that has been trained on unbiased, let's call it unbiased data. We'll see why. So in the first experiments, we simulate a manifestation shift. So this is the pneumothorax example that I mentioned at the beginning. We see that manifestation of, for example, pneumothorax but no tubes is rare, or vice versa. So that's what we try to simulate. For example, removing only pneumothorax but keeping the tubes. And what we see is that the model that was trained, basically this 93 accuracy is the happy metrics that I mentioned at the beginning. But then when we evaluate that bias model on the simulated data, we see that the performance is actually really bad. And probably because the model has learned to detect the chest drains, not actually the pneumothorax. So it's a learning shortcut. And you might be wondering, okay, is that because your generated images are really bad and your fidelity is actually really bad? And that would be a good question. But we see that when we have a model that has been actually trained on better data that is less biased, it does indeed do better with on the data set that has been augmented with synthetic images. So it's not the generative model's fault. See that it still performs much better than the weak data set. But of course, it's hard to have an unbiased data set to train with. So that's the point of this paper, right? Another example, we simulate acquisition shift. So in this case, let's say we have a data set of COVID-19, and we have positive images from one hospital, from one data set, and all the negative images are from a different data set, a different hospital. What tends to happen is that, for example, these markings saying whether side of the image is left or right are very different, and sometimes the field of view is different as well. For example, across institutions, maybe there is more space above the shoulders, etc. So these features of the images are much easier to detect for a model than whether there are opacities in the lungs or not. And so we did an experiment for this to simulate the images that were not present in the biased data set. We asked RadEdit, a model, to remove the COVID-19 from the images. So we see clear lungs, but then the rest of the features of the image should look the same. And we see the same thing, basically. The bias model does incredibly well on the biased, so the weak predictor, sorry, does extremely well on the biased data set, biased test set, but not on the synthetic. We trust, again, our model, because we see that actually a strong predictor does pretty well and generalizes to dataset shift when trained on the when tested on the synthetic data and trained on the less biased data set. Sorry, I was just looking at the questions. One last experiment is population shift. So, for example, we have a data set, let's say, for segmentation, and in these types of data sets, there are not a lot of pathologies. So a lot of the lungs are clear, easily segmentable. So what we try to do is add pathologies and see how well segmentation models do. So, for example, we to these images we had a pacemaker at the top, and we had consolidation at the bottom, and that's the result of this operation. And what we see is that so in blue we have the ground truth, and we see that weak predictor, meaning models that were trained on data sets of patients that had no pathologies, fail at generalizing to images that present pathologies. And again, it's not necessarily because the generative model does a bad job, but because when we actually find a data set that has lung segmentations and pathologies, we do see indeed that is robust against pathologies. Okay, so just to finish this part, we introduced RadEdit, a generative model that reduces artifacts compared to other models in the state of the art and avoids confounders by using different types of inclusion and exclusion masks during editing. We also have a generative model of chest X-rays, text-to-image model, can be conditioned on text, or it can also generate images unconditionally. And then we have data sets that are built from real images, can be used for stress testing of classification, segmentation models, potentially others. And then I haven't talked about that in this talk, but they can also be used for dataset augmentation, for data augmentation, to improve robustness of downstream models. So I want to thank my colleagues again. Done a lot of work on all these papers in the last months, in the last couple of years. And also, if you want to be part of this group, just want to announce that we are hiring. We have a position open in our team here for a senior researcher. And then there is a joint internship between us and the other Cambridge in New England, in the States. So feel free to get in touch or to apply directly in the websites from the QR codes. Okay. Thank you.
Thank you, Fernando. It's a wonderful talk. Now it's time for questions. We have many questions in Q&A. Would you like to answer them live? So, so after if possible, you may repeat. Yeah, I can read the questions for you. So
yeah, I can go through the questions. I see there are loads. So
yeah,
as
it's because we might upload the video, so it's nice if you can repeat the
yeah, of course,
question then answer it. Yeah,
of course.
awesome. Thank you.
Yeah. Let me know when we should stop.
Yeah.
So I have a question here saying, is RAD-DINO computationally scalable to 3D problems? I think I haven't seen yet any method using it for 3D. It's not cheap. RAD-DINO, sorry, not RAD-DINO, just Dino V2. It has a lot of optimization tricks to make it work fast, and minimize memory. So that I think is yet to be investigated. I think that's a good question. Of course, this audience is very interested in 3D. Let me see what else we have here. I see a question. The original Dino paper used transformer blocks as its core processing block. Did you do the same, or did you also experiment with convolutions? Yeah, we use transformers as well. So these every image encoder I've discussed in this paper is a VAT. Yes, even the noising unit of the diffusion model has convolution and self-attention as well, and cross-attention inside. So a lot of VATs everywhere. I see. Is the data set from one source? Do you face any domain shift problems? The data set from one source. I guess this is about Dino. So I have I put a table somewhere with the sources of the different data sets. We have about 6. We also have some models trained with private data. So for now, we haven't observed a lot of domain shift, and we do evaluate on datasets that we haven't trained on, and things seem to work pretty well. So we tend to find that these models get quite very robust because they're trained on much larger datasets than others in the literature. I see more questions about 3D. So interesting finding that simple neural architectures do the job. My question is what would it take to go from 2D images in multimodal AI to 3D scans? Is it just more compute data, or are there some interesting ideas out there? I think 3D images are a lot more challenging because of many different things. Fewer images are available, obviously much more expensive computationally. We have very different resolutions, very different image sizes. We have different reporting styles. It might be that when we look at a chest X-ray, there's less variation in the reports, whereas if we look, for example, at a CT scan, maybe we want to write a report organ by organ, or we want to do the whole thing. I think there's a lot more questions to take into account. So I think it's a challenging but very interesting area of research. I see. How do you fit image embeddings to the LLM? And the answer is using a projector. In my slide, it was called an adapter. If you look at the Lava paper, you will see more information about that. So we pass the image through the VAT that gives us patch tokens, and those patch tokens are projected to the LLM space using the adapter. Yeah, that's how it works. What else? Can we have access to pre-trained RAD-DINO soon? Yes, hopefully. What is the architecture of the adapter in MAIRA? So it's just a shallow multi-layer perceptron, an MLP. We investigated with different layers. Making it a little bit deeper tends to work better. I think there are more details about that in the paper. How do you generate segmentations using the pre-trained encoder? Right. So for evaluations, we did something very simple, which is, I'm just trying to remember. You encode the image, you have 37 by 37 latent space patch token representation. And then a couple of things you can do to make it simple. Either you just throw your 1-by-1 convolution classifier on top of that low resolution space and then just upscale your segmentations to the original image space, or you upscale first and then you classify after that. And obviously, this is not an ideal way of decoding your latency into a segmentation, but it's good enough to compare across models. But as I mentioned, we also experimented with more powerful decoders like UPerNet to see how much we could push the performance, and you really can. I can go. I can keep going. I see it's 5 o'clock here. Can you tell me what would you?
Yes. If you like, we can go through more questions.
Okay.
If you like, yeah. So participants can choose whether to stay.
Okay. Yeah, I have some time. Let's see. Let's pick up the latest question. What are the next steps? Yeah, so we planning to look at mostly what happens when you use multiple views in the LLM. So MAIRA is given multiple views of the same study or prior studies as well. And yeah, potentially working on 3D at some point. Longitudinal data, I see another question. Does the model work with longitudinal data? So this is exactly what I'm talking about. We tend to, we use current image, but the plan is to start working with prior images as well. And this is in line with our prior papers, for example, Biomedical, where we're doing contrastive learning, but using current and prior images as well. Have you considered including synthetic report from some chest X-ray dataset without reports? Yes, there are more and more papers showing improved performance using synthetic data. Obviously now we have all the tools for this, right? We can create images from reports using the RadEdit generative model. We can create reports from images using MAIRA. But I think we need to be very careful with these kind of methods because sometimes things get a little bit circular, and you might be biasing more and more your data sets. So definitely something that I think with some care could be achieved, but it's not something we have been looking into very deeply. Did you try fine-tuning the encoder? I guess this means the image encoder while we train MAIRA as well. So I would need to double check the experiments. I think fine-tuning the encoder was tried and it might have improved results, but it's more expensive, of course. But I might be wrong. I would need to check. Someone says, so Hania says, I was curious if you have explored segmentation of other structures in addition to lungs, for example, heart and bones. Have you looked at all into whether segmentation can help with the radiology report generation task? So this is an excellent question. We, for our evaluation of our image encoder, we actually tried segmentation of lungs, lung lobes, I think, lung zones, an approximation of lung lobes. But we also looked at ribs, which are more challenging, and we look at pneumothorax, which is really challenging, and we look at chest tubes, which are very small. So we look at all that, and I think the question of whether segmentation can help with report generation is very good and definitely worth exploring.
it seems we have went through most, if not all questions. So thank you, Fernando. And I think that's it for today's talk. Thank you for all the participants for joining the talk, and thank you, Fernando, for the wonderful talk.
Thanks for the opportunity. Thank you very much.
Thank you. Thank you.
Bye-bye.
Bye.
Bye-bye.